Let's continue and put on a different lens and examine long COVID at the biological level. Dr. Akiko Iwasaki is professor of immunology at Yale Medical School, and her research is focused on the underlying pathophysiology of long COVID. Dr. Iwasaki, it's a great pleasure to have you with us today.
Thank you very much. Can you see my slides?
Yeah.
Yes,
good.
great. Okay, thank you. So I'm really delighted to be on this panel and speaking now more about the biology that underlie in long COVID. So I'm an immunologist, been studying virus infections for many years. And so my take on this disease will be coming more from the immunological lens, but I'm also interested in the physiological changes that happen. So long COVID, as we've already heard from all the speakers, affects pretty much all the organ systems that we can think of. Some of the key sort of features of the symptoms of long COVID relate to the brain fog and other cognitive issues, as well as circulatory systems, immune system, the lung, and many others. So I want to start by stating that long COVID is not alone. The COVID is not alone in causing long-term diseases, that there are many unexplained post-acute infection syndromes that occur after encounter with distinct pathogens, many of which are viral pathogens that are listed here, including Ebola, dengue, polio, SARS, chikungunya, EBV, and many others, while others also include bacterial and parasitic infections. And this was a review that I was fortunate to put together with Jan Chotka, who himself have ME/CFS and have done enormous amount of research in this area. This just shows you that the underlying biology of long COVID has to be understood from the lens of these other post-acute infection syndromes that likely share a common mechanism. So there are many hypotheses that can account for long COVID. But here I listed 4 major ones. One is the viral reservoir, where the virus can remain in some tissues that replicate and cause chronic inflammation, both at the innate and adaptive immune phases. It's also possible that there may be non-infectious viral particles or defective viruses that create these antigens or RNA that can be sensed by the immune system. Second possibility is autoimmunity. This can be mediated by an acute infection with a given pathogen that can trigger either bystander cells that are reactive to self-antigens or have molecular mimicry that can detect antigens that are shared between the virus and the self. The third hypothesis is microbiome dysbiosis and or reactivation of chronic viruses, latent viruses, like the herpes virus family members. Fourth hypothesis is tissue damage that can be caused by an acute infection that is difficult to restore or repair. And there is evidence for all of these hypotheses right now. There are over 100 papers demonstrating viral proteins and or RNA, and in some cases, infectious viral particles throughout the body. And you will hear more about this from the next speaker. And so I'm not going to dwell on this too much. There's also the autoimmune hypothesis. There is a very nice paper by Jim Heath's group last year that demonstrated that lupus-related autoantibodies are elevated during the acute COVID phase, and that's one of the 4 predictive factors for development of long COVID. However, we have not found the same type of autoantibody correlation with long COVID at the later stages of long COVID. So again, pointing to what Lisa was saying, that the timing of infection and disease really does matter. Oops. The other hypothesis that I mentioned, the reactivation of Epstein-Barr virus has now been shown by at least 3 groups, if not more. The Jim Heath paper that I just mentioned also detected EBV viremia at the acute phase of COVID as one of the other 4 predictive factors for long COVID. Michael Peluso's lab, as well as our lab, have demonstrated that Epstein-Barr virus reactivation is associated in the long COVID patients. And we and others have been looking at tissue damage hypothesis, and we had a mouse model of mild acute respiratory infection with Michel Monge's group at Stanford, where we were able to demonstrate that respiratory-only mild infection can cause significant damage in the CNS, the brain of the same animals, 7 weeks post-infection, even though there is no evidence of remnant virus in the lung or in the brain in that model. So we're trying to understand how this acute transient inflammation can actually lead to this long-term consequences, likely involving epigenetic changes in the cells within the CNS. But today, what I like to focus on is our preprint that we have with Dr. David Petrino, whom you heard from the first session. We're very, very fortunate to be collaborating with Dr. Petrino's group, who treat thousands of long COVID patients, as you've already heard from him. Plus his lab members, Jamie Wood, Laura Tabakov, and Dana McCarthy, who are all part of this study that we're doing together with the Yale team that's indicated here. So Aaron Ring and David Van Dyke are co-senior corresponding authors of the study, and John, Rahul, Pei Wen, Jill, Jeff, and Sasha are co-first authors of the study along with Jamie. So what we did here is to really focus on the what David called the syndromic COVID, long COVID patients who developed long COVID in his clinic that had been assessed and treated in his clinic with a lot of great symptom surveys as well as electronic health records from the patients. What we added to that is sort of the biological analyses using multiple different strategies, such as flow cytometry to look at cell types and activation state of different immune cells in circulation. We also looked at human exoproteome reactive antibodies. So these are antibodies that react to human proteins that are either transmembrane or secreted. We also profiled SARS-CoV-2 antibody, both at the level of neutralization as well as binding capacity to different antigens. We also did peptide display library using bacterial phage display, where we can pretty much find out antibody reactivity to any linear epitope that exists. And then we did plasma proteomics to look at cytokines and hormones. So the demographic of this particular study participants were as matched as we could possibly do. Again, thanks to David and his team, we were able to recruit patients with long COVID in purple here versus people who have convalescent control, which are people who were infected around the same time in the same geographic location, but have fully recovered from COVID. So you'll see this comparison. We also included healthy controls and healthcare worker controls as comparison. And so the age is, median age is about 45.8 for the long COVID and 41.4 for the convalescent control. We have predominantly female patients, as you've already heard that there's a female dominance to this disease. And we focused on mild COVID, acute COVID cases, where we are hoping to unravel some biological underpinning within the syndromic long COVID participants. So they have very small percentage of people who were hospitalized during the acute phase. And days from acute COVID were around 400 days plus or minus, meaning that this is a much, much later time point than what others have reported thus far for biological analyses. So I'm going to give you some highlights of what we found through the multiple different biological studies that we've done. First of all, using the flow cytometry, we detected elevation in the non-conventional monocyte population. You can see that the purple, the long COVID group, have higher levels of these circulating non-conventional monocytes compared to healthy control in orange and convalescent control in yellow. And we also saw conversely, a reduction in circulating dendritic cell type 1. So CDC1s are well known for their role in priming T cell responses, particularly type 1 immune T cell responses and cytotoxic T cells. And these cells were much reduced in circulation from the long COVID patients compared to the controls. We also saw increased activated B cells and double negative B cell population in long COVID, indicating some sort of immune activation that involves B cells. And that could actually be related to what I'm about to tell you with herpes virus reactivation in these patients. With respect to T cells, what we've seen so far is that there is a significant reduction in circulating central memory CD4 T cells, and conversely, an increase in the exhausted T cell population in the long COVID patients, both CD4 and CD8 T cells. When we stimulated these peripheral blood cells in vitro to look for the T cells' capacity to produce cytokines, what we saw was quite interesting. We saw elevation in IL-2, IL-4, and IL-6 from CD4 T cells in long COVID patients compared to the convalescent control or the healthy control. And some T cells even secreted two cytokines together. The IL-4, IL-6 double positive CD4 and CD8 T cells were much elevated in the long COVID participants compared to the others. In fact, these cells weren't even found in the controls. And I'll tell you that these cells may again be related to the EBV reactivation status of these patients a little bit later on. So I just want to emphasize for the non-immunologists that these types of cytokines are normally not associated with an antiviral immune response per se. The IL-4, for example, is a Th2 cytokine, which is often associated with helminths infection or allergy. When we examine the long COVID patients' antibody response to SARS-CoV-2 antigens, what we found was that there was an elevated response to anti-spike antibodies in the long COVID patients. And we control for the number of mRNA vaccines that people have received prior to the recruitment into the study, because we wanted to make sure that it's not the vaccine doses that are resulting in the changes in what we see. So interestingly, anti-spike, anti-S1 region of the spike, and anti-receptor binding domain of the spike, all of these antibodies are elevated in the long COVID patients compared to the controls. However, these antibodies appear to be poorly neutralizing. So what does this all mean? We don't really quite know yet, but it could mean that these antibodies against the spike protein is being elevated as a result of persistent antigen driving these antibodies, and yet functionally these antibodies are not very, it's poor in neutralizing the antibodies in the viruses. When we examine the circulating cytokines and hormones, the number one and the most different factor we detected in the long COVID patients was cortisol. So this upper panel is really showing you the negative log 10 p-value for all the different factors that we analyzed among the different groups. And what we saw was the cortisol level was actually quite significantly different between the healthy control and the convalescent control. And the long COVID patients had about half the level of circulating cortisol compared to the controls. So normally cortisol is a very important diurnal hormone that is regulated through the hypothalamic pituitary adrenal axis gland axis. And adrenal gland produces cortisol, and when that level is low, the pituitary usually produces this ACTH hormone in order to kind of elevate the low level of cortisol. However, we're not seeing this increase in ACTH level in circulation, suggesting that perhaps there's a pituitary or even hypothalamic dysregulation in long COVID patients, resulting in this lower levels of cortisol. And we also ensure that the time of collection during the day is not significantly different between the cohorts, because as I said, this cortisol is a diurnal hormone that is peaks in the morning and it trails down during the day. And we wanted to make sure that the time of day was similar between these cohorts. What about these other hypotheses? Well, we looked at latent virus reactivation by looking at the antibody levels in patients with long COVID. And what we found was an elevation in IgG that is reactive to the glycoproteins from Epstein-Barr virus. So the two different glycoproteins was detected to be elevated in antibody reactivity in the long COVID patients compared to the controls. We also saw a similar elevation in the VZV reactive glycoprotein E reactive antibodies in the long COVID patients. And this is not because there was a seroprevalence difference of EBV in long COVID versus non-long COVID, because when we examine that using the latent chronic state of seropositivity in these patients, there was no difference between the long COVID and the control groups. And there was very little mononucleosis evidence in these patients either. So it's really the recent reactivation of EBV that is detected predominantly in the long COVID patients. So this is another, we used two other orthogonal sort of approaches to look at EBV reactivation. So the original data I showed you is using the REAP technology that Aaron Ring Lab has produced. But we also used a bacterial phage display library to examine the linear epitope indicated here, reactivity of antibodies from long COVID patients. And that was also elevated significantly over the controls. The BZVGE reactive antibodies were also elevated in the long COVID patients. And as I alluded to earlier, the strange IL-4, IL-6 double positive CD4 T cells that we saw elevated in the long COVID patients, that level on the y-axis actually correlated very nicely with the EBV reactive antibodies scores for both the P23 and the GP42 epitopes. So again, what this may be suggesting is that there is a reactivation of EBV that corresponds with this sort of Th2 type of CD4 T cells that is being elevated. And it's well known that the GP42 of the EBV envelope actually interacts with the MHC class 2 molecule to interfere with the productive Th1 induction, which is able to clear the virus. So the virus prevents that and usually induces this type 2 type of CD4 T cells. And maybe this correlation is telling us that there is a biological link between the Th2 skewing and the EBV reactivation within the same patients. Okay. So what about autoimmunity? Well, during the severe acute COVID phase, we have demonstrated that there are numerous functional autoantibodies that develop in patients that actually predispose patients to severe disease. However, when we looked at these autoantibodies against human exoproteome using the REAP strategy that the Ring Lab developed, we did not see a significant difference in the number of autoantibody reactivities or the number of reactivity as compared to the long COVID propensity scores. So we haven't seen a clear evidence as we saw in the acute phase of severe COVID. The same kind of evidence is not found using this strategy. And we are still open-minded about the autoimmunity as being part of the long COVID pathogenesis because we have not looked at intercellular antigens or T-cell immunity against the self-antigens. But at least the exoproteome reactive autoantibodies are not significantly different. So taking all of these, we measured thousands of different parameters from these patients and applying a machine learning to in a matched cohort. So we matched for age, sex, and hospitalization status for the long COVID and non-long COVID, and we did machine learning analysis. We were able to distinguish long COVID and non-long COVID based only on the immunophenotyping from these patients. With a 94% accuracy. And we were also able to discern that the factors that are kind of contributing to this distinction of long COVID is not autoantibody epitopes, but more to do with the SARS-CoV-2 specific antibodies and EBV reactive antibodies, circulating immune cells, and the hormone, the cortisol that I mentioned. So altogether, these factors contribute to our ability to distinguish long COVID with 94% accuracy. And the factors that really contribute to the differences between long COVID, some that are lower in the long COVID and others that are higher in long COVID, are listed here in this GLM classifier estimate. And what we find is that cortisol, again, is really stand out as a number one factor that is a distinguishing factor for long COVID. But then there are other very important factors that I've already mentioned, such as lower level of circulating central memory cells, CDC1s, and then CD8 T cells are among the ones that are found to be lower in long COVID. And that is contributing to this machine learning algorithm that allows us to distinguish long COVID versus non-long COVID. What's higher in long COVID is also interesting. Things like the EBV reactive antibody is certainly one of those factors. Galectin-1, which is another factor that we isolated from circulation, as well as TGF beta family members that are elevated, and CD4 T cells and sort of Th1, CD8 T cells. So this is sort of what we're seeing in this sort of particular long COVID study. And of course, a lot more results are needed to confirm these findings, but we are starting to see some biological hints of what's going on in these patients. So just kind of going back to the hypotheses that I mentioned already, the viral reservoir hypothesis still stands as a potentially important candidate. As I mentioned, there is this elevated antibody levels and sort of this exhausted T cell features that are suggestive of a viral reservoir. And of course, others have demonstrated, for instance, David Walt's group, demonstrating that circulating spike protein is found in long COVID patients months after the acute phase. So based on that, we are starting a clinical trial called Paxlovid LC through the Yale Lesson study with Harlan Krumholtz and others. And we just got an IRB approval to recruit patients. We're also still quite open about this reactivation of EBV and potentially dysbiosis, which we didn't study in our current study. Tissue damage is something we're investigating using various animal models. And then autoimmunity, as I mentioned, there may be a T cell component, things that we haven't studied yet, but it's something to be really studied deeper. So quickly, what we need to do now is really dive deeper into the biology and underlying pathogenesis that drive this disease, including viral reservoir, autoimmunity, latent virus reactivation, and tissue damage, that's resulting in very common features such as vascular damage, microclots, platelet activation, hormonal imbalance, and mitochondrial dysfunction, and which then leads to a constellation of different diseases and symptoms that are listed here. What we need now is not just more research, but looking at clinical trials, randomized placebo-controlled clinical trials to inform us, who's benefiting from what type of therapies, and can we learn biomarkers from those studies to target patients for better therapy. So I'm going to end here by thanking all the people that are involved in this large number of studies. I was able to mention some of the key players here, but there are so many others that I didn't have time to thank, but I'm going to end here. Thank you.
Thank you very much, Dr. Iwasaki, for your work and for being such a prominent voice on behalf of patients. We appreciate it.
Thank you.
Our next speaker, Dr. Van Elzeker, is a co-founder of PolyBio, a very unique nonprofit research center. Dr. Van Elzeker, thank you for being with us today, and thank you especially for filling in for Dr. Prohl on such a short notice. Thank you.
Thanks, Ovid. I appreciate it. So, yeah, I'm really glad to be here. Thanks for inviting me. And I'm just going to walk through a little bit of some, mechanisms that may be driving long COVID, sort of following up Dr. Iwasaki's talk, and talk about maybe a little bit about how we might address them. So next slide, please. So, I think when long COVID first started becoming this phenomenon, the patient-led research consortium really was an important voice early on in pointing out, hey, there's something going on here. You know, I had COVID and I'm not better. And it took the world by surprise in a lot of ways, or at least a lot of the world. I think for a lot of us, it unfortunately was not surprising. You know, we've been working with MECFS for many years, and this is an example of, you know, I don't like to use the word post-viral because that's a really big question. Is it actually post-viral? But, you know, post-acute infection conditions in which people don't fully recover. You know, it tends to be, we've talked about this in other talks earlier today, you know, long COVID or even PASC are kind of syndrome diagnostic labels. It's very broad and heterogeneous. Practically speaking, in the research world, that means that there's some important considerations when we talk and think about it, and it's been addressed a little bit earlier today as well. But it makes group statistics difficult because you may have something that's particularly important in an individual, and that might get washed out in a case control study. So I think it's important to keep in mind the heterogeneity of this condition. Larger studies are better, the type of study that Dr. Iwasaki and Petrino did with a relatively large cohort, so that you can pull out potential patterns. But again, sometimes those are driven, let's say 15% of the cases have some abnormality and none of the controls do. That's probably going to be enough to drive a group statistic finding. So it's important to think about that. Within subjects, challenge studies are really valuable. So if you can take an immune cell from a case and from a control and challenge it in some way, that eats up some of the variance in your studies. Talking about a study of long COVID or PASC is almost like talking about a study of cancer or of people who have a broken bone. You know, there's a big heterogeneous group here that needs to be sort of considered. Easily measured analytes are going to get preferenced. So if something can be easily measured in blood, there's going to be 1,000 studies on it. If something is difficult and can only be measured in tissue, there'll only be 1 or 2 studies on it. That shouldn't be a measure of the importance of the finding, but it is a fact of how the research is going to play out. And you know, in general, I think personalized medicine is going to be an important approach. Next slide, please. So this was a study early on to talk about the concept of viral persistence. Germany has a law on the books where if there was a new pathogen that emerged, they had the ability to do detailed autopsies, even without consent from the families, although I think they ended up getting consent afterwards. So this was an important early German study that was very systematic. They got a lot of different types of tissues early on. And here is an example of them finding a viral antigen in the medulla oblongata cranial nerve associated brain tissue of someone. Now, remember that this is someone that died of COVID, so it's spread quite far. Next slide, please. This is a more recent study that just came out. It had been in preprint for about 8 months. Something like that. But Daniel Chertow's group at the National Institute of Health, they found widespread viral protein and RNA, including in some cases ability to culture, and up to 200 days after initial onset. Now, again, these are people that died, so it's kind of the worst case scenario. We don't know that this is what's happening in long COVID, but it's a conceptual point being made that this is a widespread virus that can infect lots of different tissue types, including the brain. Again, it's worst case scenario because these people die. But importantly, this is a study that really prioritized a short death interval. They got all of their tissue within 22 hours. That's a very, very difficult thing to do. You know, sometimes someone can die at 2 in the morning and you have to be able to be reactive to that. So when we're looking at these kinds of studies, that's important to keep in mind. And also the preservation of tissues is really important to keep in mind. If you're looking for protein or if you're looking for RNA, those tissues are preserved in different ways. You can prioritize either of those things, but it's difficult to prioritize them both. So, you know, I think it's important when we look at these sorts of tissues in an autopsy type study. Okay, next slide, please. So kind of moving into people that did not die, several researchers took what was essentially a convenience sample and just looked in tissue of people that had previously been infected with SARS-CoV-2. This is a study in the BMJ that identified SARS-CoV-2 nucleocapsid protein and viral RNA in colon, appendix, ileum, hemorrhoid tissue that was being removed, liver, gallbladder, and lymph node, collected during surgery from patients 9 to 180 days after acute COVID-19. Again, this is not necessarily long COVID or PASC. We don't really know. Is this, the reason that the hemorrhoids flared in this individual with so much that they asked for surgery? Those are sort of important questions. But again, it's showing that the virus is capable of persisting. So next slide, please. And even next slide, please. So that was a study by Gabler and Sar Mahandru from Mount Sinai. It's a whole group there. And again, it's somewhat of a convenient sample where they found viral reservoirs in the gut after COVID-19. Here they're showing SARS-CoV-2 nucleocapsid. Importantly, with these studies that are trying to demonstrate the possibility of viral persistence in tissues, in some cases, evidence of virus is in certain fractions that are more easily sampled. So for example, you might find it in stool, but not in a nasopharyngeal swab when you get, a couple of months out. There are several examples of that in the literature of, unfortunately, the virus doesn't really care about our study designs. And it would be really great if everyone with a persistent virus had an easily detectable nasopharyngeal swab, but that's not how it works. So for example, there was a tragic case study of someone who, there's a stillbirth, and the mother tested negative by nasopharyngeal swab, but the placenta tested positive in that case. Viruses are obligate intracellular parasites. They need to be in a host cell to do anything. They don't metabolize on their own. They don't reproduce on their own. So their propensity is to go inside of cells. And for that matter, their propensity is to go inside of tissues in most cases, because in the blood or in circulating fluids, they're really vulnerable to being attacked by an immune response. So getting into tissues, I think, is harder. You're going to see fewer studies doing that. You're going to see much more frequent studies looking at peripheral inflammatory markers, which can be really important. But one of the things that we've learned from the MECFS world is to be a little bit careful. Just because lots of studies show inflammation in the blood, that doesn't mean that inflammation is the disorder, that is the basic pathophysiology, just because we've seen it in a lot of studies. Sometimes there's a reason for the inflammation. So next slide, please. So this is a study that's a little bit more into like PASC proper. One of the sequelae of COVID-19 is erectile dysfunction in some people. And this is an example of 2 case studies in which that was one of the sequelae of acute COVID. And during surgery to address the erectile dysfunction, they decided to look and found evidence of viral particles in the penile tissue. Again, not necessarily a nasopharyngeal swab or blood. Next study, please. Sorry, next slide, please. So this is a study from a group at Mass General. Alessio Fasano and Lael Yonker and Brittany Borabong are working on this, and they are studying something that is an analogy to long COVID. It's a type of sequela of COVID-19 called MIS-C, MIS-C, so a multisystem inflammatory syndrome in children. And these are kids that may have been asymptomatic. They may have had a relatively mild case, but then a few weeks after that, they have this incredibly robust immune response that comes out as a later consequence that in some cases has killed kids. It's really tragic. There's a story kind of that we should just be able to let it rip in kids because they're usually mild. They are usually mild, but when it's not mild, it's not. And this is also evidence, importantly, that at least in some kids, for reasons that we don't completely understand, maybe there's a vulnerability, but maybe not, the virus seems to stick around in a gut reservoir and then can, again, for reasons that are not completely understood, can reemerge or at least drive an inflammatory response weeks later, and in some cases quite severe. Next slide, please. So PASC proper is kind of something that we really need to focus in and study more. From my perspective, especially tissue biopsies are an important priority, but this is an example of a study that Dr. Iwasaki mentioned briefly from David Walt's group at Harvard Med. So David Walt is one of the inventors of the Quanterix and Illumina panel of devices. So they have a Simoa technology that can measure in very sensitive low amounts circulating spike protein. And in this study, they studied 62 individuals, 37, all of whom were post-COVID, 37 of whom had PASC. All but 2 of the PASC cases, or long COVID cases in this case, were mild, so only 2 were hospitalized. And they found some sort of protein, whether it's spike S1 or nucleocapsid, in about 2/3 of their long COVID cases. In all 12 of the individuals with long COVID that they had longitudinal data for, they found it at least 2 time points. They took that as, quote, the presence of circulating spike in PASC patients up to 12 months post-diagnosis strongly suggests that SARS-CoV-2 viral reservoirs persist in the body. So sort of Occam's razor is not that these proteins are sticking around and surviving proteases and surviving sequestration by immune cells, but rather that they are being produced de novo by reservoirs. Next slide, please. So the microclot phenomenon is something that I think a lot of patients are familiar with. Largely, Reese Pretorius and Doug Kell have done this work, and they've been doing it in other conditions for many years. So they're really experts in this topic. It's a method that is not yet clinically available, so you can't get it ordered by your doctor, but it is a relatively easy assay for research groups to collect plasma. You can use banked plasma. There's an optimal way to do it with citrate platelet poor plasma, but you can use it on cases and controls as long as they were collect similarly. These are clots that are somewhere between 20 and 500 micrometers in diameter, and they're just incubated with THT or theoflavin T, which shows the fibrin amyloid nature of these microclots. And it's different than D-dimer, which is clinically ordered. It's not necessarily the case that D-dimer would be elevated, although Linda Gang, for example, from Stanford reports that somewhere around 15 to 20% of her long COVID cohort shows elevated D-dimer. D-dimer is released when clots break down, and the whole problem is that these aren't breaking down. Next slide, please. So this is an example of a microclot done at my own lab, along with Lael Yonker and Brittany Borabong. Again, somewhere around 50 micrometers. Given that the smallest blood vessels, the capillaries in organs, are about 50 micrometers in diameter, you can see that that might cause some diffuse, somewhat idiosyncratic problems in different organ systems. Next slide, please. Here's an example of an abnormally shaped microclot, and we've seen a couple that look like this, where we think maybe it's a piece of vasculature or something that was pulled off. We're not sure. Next slide, please. So small fiber neuropathy is something that's been shown in a very small study, very small, in about 2/3 of people with long COVID in this small study. Whether this is related to the microclots or whether it's some sort of an other, pathophysiology is important to address. And it's being addressed in a clinical approach with IVIG in a small clinical trial right now with Peter Novak. Next slide, please. So this is from my own group, and I guess you're sort of seeing it for the first time. So this is 12 individuals. We deliberately recruited people with neurocognitive problems. So we asked for people who had trouble concentrating or abnormally strong fatigue. And in a paired samples design, they're compared to 12 individuals that were scanned with the exact same scanner and the exact same protocol pre-COVID. And what we're seeing here is an increased uptake in the posterior cingulate cortex. When we dig in and look, this is again group statistics. When we dig in and look at an individual level, there's individual patterns of activation. So what this is showing is the innate immune cells of the brain, the glial cells, are activated. So it's a type of a neuroinflammation. It doesn't necessarily mean penetrating immune cells, but it means that the glia are activated. So, this is probably related to the fact that this is an area of the brain that is particularly densely vascularized. It may be related to, perhaps microclots or vascular damage. This was paid with our own fundraising, and we're trying to get funding to expand it. Or actually, we're able to expand it with our own fundraising as well. So we don't know if it's perhaps, in a different analysis, we showed that it was really centered around thalamus, which is where there's a lot of circumventricular organs. These are areas where there is kind of a window in the blood-brain barrier. So maybe inflammatory markers are able to make it through that and cause some damage and inflammation. So this is a type of neuroinflammation shown in long COVID. Okay, next slide, please. And, I just want to drive home the point that, one of the reasons that one might see neuroinflammation is direct damage to vasculature. That's possible. But another is that the brain is in constant contact with the rest of the body and receiving inflammatory signals through the vagus nerve. So this is a paper that I wrote many years ago. I'm not at all saying that everyone with these ongoing symptoms has an infection of their vagus nerve. It was a description of a mechanism that I think is important. So, when we talk about cytokines, they're going to be found in a lot of studies. Those are protein molecules that are lipophobic. They don't easily cross the blood-brain barrier just in an endocrine fashion. So what happens is that the vagus nerve detects them in the periphery and sends a signal up to the brain saying, hey, brain, your body is sick. There's inflammation down here. It triggers inflammation on the brain side of the blood-brain barrier. And this is a study here, not about COVID per se, but just talking about the phenomenon of the vagus nerve detecting gut dysbiosis. So if that is one of the issues that may be happening in individuals with long COVID consequences, then that is the mechanism by which it's detected. So I think I'm about at time. Next slide. Thank you very much for everyone on my team and for our study participants.
Thank you very much, Dr. Van Alzekar. And just on your last slide, I think there were recent studies on the microbiome changes in ME/CFS, which may very well be relevant here. So thank you for this wonderful presentation. And I think we only have time for one question. There were many questions for Dr. Iwasaki and Dr. Van Alzekar, but Dr. Iwasaki, maybe one question that came for you specifically. Are you able to monitor the changes in the immune response over time? And the second question is, do you see a change in the response of males versus females? And should that be factored in the design of clinical studies?
Yeah, excellent questions. Thank you. So the study that I presented today is a cross-sectional study, one time point, but we are starting other studies where we can do a longitudinal analysis from the same patients over time. And looking at various different published studies, there appears to be immune signatures that keep shifting over time. So our study is much later, but if you look at 3 months, 4 months, 8 months, the signature seems to be different from what we're seeing. So I think there is an evolution of immune signatures that change over time. And with respect to the sex differences in symptoms and immunology, we are seeing very distinct differences between male and female patients. And that's something with respect to both symptoms prevalence as well as immune signatures that underlie these diseases. And I think that's a very important question of having to really pay attention to the sex differences in the immune responses and trying to come up with better therapies and diagnosis.
Wonderful. Thank you very much. So I think we're right on time now to take a break. I really wanted to thank all the participants, the speakers this afternoon. Really appreciate it. And we will be taking a 10-minute break now.